First of all, I would just like to thank Tim and his team for putting together a really great audience and esteemed audience at that, and allowing me the opportunity to come and discuss Innova's technologies. I'm Brian Carpenter and I'm Innova Biosciences Sales and Marketing Manager. Next slide please, James. One thing is I'd like to just give an understanding of what our core competency is, what is our main focus for the company, and then sort of the problems associated with traditional bioconjugation and the solutions that we provide in regards to Innova's bioconjugation technologies. And then I'd like to just give a brief video demonstration which shows quite nicely how simple the process is in regards to the bioconjugation process. And then to show some performance data of our conjugates, of conjugates using our technologies, and sort of partnering opportunities that we're interested in from either yourselves or demonstrated through existing partnerships that we have available at this point in time. So next slide please, James. So maybe, you know, one of the things is I think it's very important to involve the audience. So maybe we could have a demonstration of what Innova is about. So if you could talk to one of your partners and give them a handshake and just introduce yourself.
Hello, James. I've never seen
So that's what Innova is about. You know, our strength is joining two molecules together, is providing those linking technologies which stick together, work together, and create a molecule or a hybrid which allows enhanced performance. The one thing about Innova is we have a real strong knowledge base which allows us to tap into some innovative linking technologies, which therefore allows us to produce conjugates with enhanced performance. And when I say enhanced performance, I mean in regards to stability. I also mean in regards to the sensitivity in the assay of choice. We have lots of data, lots of people coming to us with problems, and we use our innovative linking technologies, which allows us to generate a conjugate, which therefore produces an enhanced sensitivity in the assay of choice. And this is really important not only to the diagnostic industry but also the academic industry, where they are pushing the thresholds of the limits of detection. Next slide, please. So let's just, sorry, I hate podiums, so I'm just going to walk around. One thing is that about conjugates is, you know, very time-consuming process. Conventional labeling methodology can take from hours up to days. Not only do you have to be aware of this time constraint, you have to be aware of the various chemistries that can be targeted, whether it be the amine groups targeted via yourselves, the carboxyls, the sulfhydryls. It's a very time-consuming process to actually identify to define the ideal chemistry. You also have disadvantages, as in you have to buy capital equipment such as separation columns, separation machines, which results in loss of material because you have, due to the separation steps, with any separation process, you're going to lose your end product or lose end material. And also one of the major things is it's scaling up. To take your products from micrograms from the R&D stage right up to the manufacturing process is not exactly the same as what you would do at the bench of what you would do in a manufacturing setting. So there can be issues with these types of technologies, with these types of approaches, and therefore that's what Innova is about. We've taken these processes, we've taken the chemistry, we've taken the scalability issues, and we've put it into an easy-use format, which allows the customer to without having to understand all these processes, to make a superior conjugate. Next slide, please, James. So a bit about Innova's technology. You know, we provide sophisticated technologies, and what I say is that we provide all the chemistry, all the various labels that you would be interested in, and we've taken away the complexity of the process. There's no need for any separation steps or dialysis steps, which allows you to maintain 100% of your recovery. One of the nice things about the technology is it's a stable covalent bond between the label itself and the antibody or the protein of interest. And therefore that allows you to store your conjugate for as long as required. At that point in time you have to question, well, how stable is my antibody rather than how stable is the conjugate itself, because it's a covalent bond. In regards to the scale process, you know, one of the things about Innova is we've taken that on board. We realized that people are now going to want to take, because our process was so simple, They want to take it from the R&D right into the manufacturing process, and that's what we geared. We've now been able to generate conjugates from the 10 microgram scale right up to 1 gram plus, depending on what's the interest of the customer. We can tailor design our manufacturing processes in-house to accommodate what the customer needs. And therefore, by having such a strong, such a strongly manufactured process or coordinated approach, we eliminate batch-to-batch variability, which again is one of the more important things in regards to the manufacturing process. And what's really important is our conjugates have demonstrated time after time enhanced performance compared to the conventional methodology. And these are our technologies: Lightning, Innova, and Thunderlink. Basically, they are labeled. In this range we have over 40 different labels, which includes the various ones such as the enzymes, fluorescent proteins, fluorescent dyes, biotin, streptavidin. Generally, all the common labels are available in the Lightning range. In the Innova range, we've now just started going into this technology. This is really geared towards the diagnostic market, but we've also actually demonstrated through our sales history that surprisingly we're having a lot of interest by the academic market in these particles, these conjugation particles. And what's the nice thing is this is really it's taking approach which the standard scientist was unable to use, and allows them to generate a conjugate using our Innova technology quite cost effectively, and then opens up a whole new customer and a whole new opportunity for the customers themselves or the scientists. And Thunderlink is what we sort of class as the next generation. This is where we conjugated oligos to antibodies or any type of biomolecule that might be of interest. And the real value is there. This is where people want to push the limits of detection and want to go beyond the picograms. They want to go towards the femtograms and beyond. And this is where we're finding a lot of interest in this technology. Only launched at the beginning of February this year, but already we're getting a lot of interest. Next slide, James. So maybe I could now show you how easy it is to generate a conjugate using Innova's conjugation technologies. And this is one of our newest technologies, which I just highlighted, which is the Innova Gold. A nice demonstration that we pulled together. So I'll just wait for the introduction to go pass by. So it's a gold nanoparticle conjugation technology. So the real value is it's to be able to work with these gold nanoparticles, which have been very difficult in the past. So here we have a scientist, a bench scientist. Here's their antibody solution. Here's their gold conjugation kit. This contains all the chemistry. This contains all the nanoparticles themselves. All they simply have to do is rehydrate the solution using the antibody solution. And what we have here is the Innova coat, which allows the particles to become conjugation friendly. Therefore, due to this technology, we allow it to be an ultra-stable covalent bond. And it's a 15-minute process, a 15-minute incubation, and that's your conjugate made. So that's pretty spectacular compared to what it is. In the past, you have to go around several rounds of optimization, where you have to alter the pH, the antibody concentration, and the buffer composition. Whereas with our kits, that's unnecessary. So you can stop the presentation. With our kits, that's unnecessary. All the customer has to do, it's all optimized already. All they have to do is take their antibody and add it to the freeze-dried powder. Sorry, by the next slide, James. And just to sort of highlight the value of the conjugation chemistry, not only let's summarize: simple technology, very easy to use, takes away the complexities, very cost effective, opens up a wealth of opportunities to the customers. What's even more important is the sensitivity. Everyone wants to always push the limits of thresholds of detection. Well, with an Innova conjugate, that is exactly what we've done. Due to the linking technology being sophisticated and being innovative, it actually allows it's not as harsh on the antibody as conventional approaches, and therefore produces an enhanced product. And here we have is just a standard ELISA assay where we've taken an antibody either pre-conjugated or generated using lightning link, and what we've actually seen was an enhanced performance of the lightning link conjugate itself. On top of that, we also see that we also demonstrate increased sensitivity. Here we're decreasing the amount of antigen in the well, and again, Innova's technology, the lightning link conjugate outperforms the standard labeling method. Next slide, please, James. So the real value is, this is, we started, or Nick started sort of the lightning link products in 2006. I joined the company in 2009, and we've grown the company rapidly from that point in time. There was 3 of us to there's 20 plus now, benefiting the company, and we're really starting to get our products to market. But also we're now benefiting from people who have used our products at the R&D stage and are now advancing to the manufacturing, using the products in the manufacturing process. We have lots of live ongoing examples of customers who've partnered with us, and they've been so happy with us, they actually were delighted to allow to do a joint press release. And this is one of the real values of the customers. One of the things is you've gone on about people wanting antibodies and how customers can add value to or suppliers can add value to their portfolio of antibodies. With our technologies, because of the simplicity of the process, because there's not much knowledge needed or expertise required, what a customer or what an antibody supplier can do is they can take our 40 plus labels and cross-reference it with their antibody portfolio. The customer says, I want that antibody with that label, can you produce it? With our technology, with our product offering, from the customer placing the first order right through to you getting the order from Innova, placing the order, we dispatching it to you, you generating the conjugate in-house, and then sending that conjugate to the customer can be 5 working days. So the real power of that is, from the antibody supplier's point of view, is you do not have to maintain stock, your stuff is not going off in your fridge or freezers, you have a demand, the customer demands the product, you have the money sitting, or the purchase order sitting in your account, there's no risk on your part. The only risk that the antibody suppliers incur is their marketing expense to raise that awareness. We're already done this with Stressmark, which is a Canadian company, and already he's having he's generated a significant amount of sales from that, and I'm sure he's very happy to support our services, and I'm sure he'd offer a testimony to us, no doubt, no problem. The other thing in regards to manufacturing, as I said, people do use the products in the microgram level, want to progress up to the gram levels. And we've done this now with 2 companies, Belgian Evolution and Immunoreagents. Again, these are 2 companies, antibody suppliers. They were using the standard labeling methods. They weren't happy with what they were seeing, so what they decided was, we needed to source a more easy process. They came to Innova. We provided them with the kits. It showed that they enjoyed the results and they signed up to the out-licensing or the in-licensing deal from them to us, from us to them. So that's just 2 examples. We have many more of these examples ongoing, but unfortunately, due to confidentiality reasons within the terms and conditions, I can't disclose them. But I'm sure that there's plenty of companies out there. The common suspects are on our list of customers. And just to sort of summarize, in custom services, we also provide antibody fragmentation conjugation services for site-specific labeling, orientation of antibodies, and the various fragments required. Also, those people that have troublesome conjugates, we realize that a lot of people have assay issues with the conjugates going off in the assay of interest, whether it be storage at fridge or freezers. They're looking for stable solutions. We're able to provide those services as well. I think this is one thing that we're going to see a lot of demand of coming in the coming months to years, is the oligo-conjugation. We are getting a lot of interest, and we are seeing this as an emerging opportunity. And we continuously identify problems that's happening in regards to the bioconjugation market, and we continuously provide solutions to those customers. And one thing I would like to say is we are an ISO 9001 and 2008 accredited facility, so that makes sure that our quality management systems allows us to make sure that we provide a top quality system to our customer base. And there, I'm just going to end in there because I think that's my 10 minutes up. But I'm more than happy to take questions in regards to our technologies or anything that may be of interest to anyone.
How do you guarantee sort of 100% antibody recovery?
Well, the nice thing is, is that we don't actually, there's no separation steps or dialysis steps in the protocol. Once the conjugate's made, and because there's very free label left over, and because of the high efficiency of the conjugation process, there's no need to do any separation steps or dialysis steps. It's a perception that people are used to doing, but due to our technology, that there's no need to do that, and that's why we can guarantee 100% recovery, because there's no need to use the materials through these processes.
So there's no sort of unbound label knocking around there?
I can't say that. It's going to, generally there's very few label left over. As long as you stick with the protocol and follow the instructions, which are very simple, there should be very unbound label, very minimal amount of it. Yeah.
Can you control the incorporation ratio?
We can, not through our kits at the moment, but through our custom services we can provide that.
Any other questions for Brian? Thanks, Brian.
Okay.
That was